Hello everyone, good morning. Welcome to Q&A class. My name is Dr. Meghna Garg and I'm biology faculty at Q&A. Students, am I audible?
Yes,
Can any one of
ma'am.
you?
Yes, ma'am.
Okay, Kamalan, thank you. Thank you for responding. So quickly, let's begin the class. So we are going to talk about Golgi body and lysosomes today. And in previous class, we had already covered endoplasmic reticulum. So do you people have any doubts from endoplasmic reticulum? Any doubts?
No, ma'am.
Okay, then quickly tell me that which cell organelle is responsible for synthesizing lipids. Write your answers in the chat box. Which cell organelle is responsible for synthesizing lipids? Yes, Kevin, Samagna, Kamalan, everyone. Which cell organelle is responsible for synthesizing lipids? Okay, Kamalan, very good. Samagna, Kevin, which cell organelle is responsible for synthesizing lipids? Shaktivel, smooth endoplasmic reticulum. You people haven't revised this? Okay, and what's the function of rough endoplasmic reticulum? Very good, Kevin.
Synthesizing proteins.
Very good, but type it in the chat box. Yes. Write your answers in the chat box, Kamalan. Kevin, Shaktivel, quickly. What's the function of rough endoplasmic reticulum? Students, answer quickly. What's the function of rough endoplasmic reticulum? Everyone answers in the chat box. I cannot see anyone typing it. Okay, Kamalan, you have written. Kamalan, can you be more specific? In previous class, I had told you this, that you need to be more specific with this term. It is involved in. So particularly protein synthesis is done by ribosomes. Since ribosomes are present on the surface of endoplasmic reticulum, that's why, particularly on the surface of rough endoplasmic reticulum, that's why we say that endoplasmic reticulum is involved in the synthesis and transport of proteins. But what do we need to remember here is that rough endoplasmic reticulum is actually not synthesizing proteins. Rough endoplasmic reticulum has ribosomes present on its surface, and ribosomes are synthesizing proteins. Rough endoplasmic reticulum is basically modifying the proteins. It is folding the that the protein becomes functional. Okay, so that you have to specifically write. Okay, so is everyone okay? Everyone, is it clear to all of you? I hope you people have understood the structure and functions of endoplasmic reticulum. Okay. Shaktivel, Samagna, you people are not responding today. Why? Okay. Now, moving ahead. So today we are going to talk about Golgi apparatus and lysosomes, and you can expect one question from here. In today's class, we are going to talk about Golgi apparatus and lysosomes. And student, let me tell you one more thing. Basically, there is a term which is called as endomembrane system. There is a term which is endomembrane system. So endomembrane system is basically the term which is used for the several cell organelles whose functions are related with each other. So you have a set of cell organelles present inside the cell which have their functions related with each other. They work in collaboration with each other. Those cell organelles are endoplasmic reticulum, Golgi bodies, lysosomes, and vacuoles. So these four cell organelles, they together constitute this endomembrane system. Why? Because the function of all these four cell organelles is related with each other. Golgi bodies, we are going to cover it in today's class. Okay, that's our outline for today's class. So right now I just told you that if we see all these four cell organelles, then these four cell organelles come under endomembrane system. Why? Because these 4 cell organelles, they have their functions related to each other. They work in collaboration with each other, and that's why we give them a term which is called as endomembrane system. Okay, understood? Understood, everyone? This was the question which is even asked in higher examinations, that the cell organelles which constitute the endomembrane system. Is it okay? Students, respond. There is no energy. Show me some energy. You people are not responding. Why is there so much lethargy in the class today? Okay, see this endomembrane system. Endomembrane system basically is the group of cell organelles which work in collaboration with each other. So endomembrane system consists of 4 cell organelles: endoplasmic reticulum, Golgi bodies, lysosomes, and vacuoles. These 4 cell organelles, they work in collaboration with each other, or their functions are related to each other. Okay, so that's why we have given them a term which is called as endomembrane system. Okay, Shubhranshu? Okay, now let's move ahead. So now we will discuss the first cell organelle, which is the Golgi apparatus. So this term particularly was given by Camillo Golgi. He first of all noticed, or he first of all worked on this Golgi apparatus. Okay, and he was a German scientist. So the name Golgi apparatus or Golgi body is after the name of this scientist, which is Camillo Golgi. Now, so this was the scientist Camillo Golgi. Now, what exactly is Golgi apparatus? So basically, Golgi apparatus comprises of membrane-bounded vesicles which approximately they lie parallel to each other. Okay, so you can see from here, this is endoplasmic reticulum, the cream color structures which you can see from here, whereas this complex structure which you can see from here, this is your Golgi body. Okay, now let's see this Golgi body. So these are membrane-bounded vesicles. Okay, so these are vesicles, and these vesicles, they are stacked onto each other. They are stacked onto top of each other like this, and inside these vesicles you have enzymes present. Okay, so different enzymes are present in these membrane bounded vesicles, and these membrane bounded vesicles, they are called as cisternae. Okay, so what's the individual unit of Golgi apparatus? So in Golgi apparatus, you have several membrane bounded vesicles. These vesicles are called as cisternae. These are flattened membrane bounded vesicles, and these cisternae, they lie on top of each other. Okay, and they are filled with different enzymes. Now, Golgi apparatus function. So Golgi is basically considered as the postman of the cell. Okay, Golgi is called as the postman of the cell. Why postman? So basically, see, this is the cell. Okay, if something has to be secreted out of the cell, then that will be packaged and modified by Golgi apparatus. Okay, so it helps in the packaging, and it is considered as the packaging and forwarding department of the cell. Why? Because whenever something has to be delivered out of the cell, and even also if something has to be delivered inside the cell, then also Golgi apparatus plays a very, very important role. Okay. Now, how does it play a role? What exactly happens? Let's see this. So initially what happens, students, in previous class, I had told you about endoplasmic reticulum. Okay. And I had also told you this, that in endoplasmic reticulum, the proteins, they are being made functional. Proteins are made functional. Okay, but how they are made functional? They are made functional by their modifications and by their folding. Okay, so that's how a protein is being made functional inside the endoplasmic reticulum. Now let's say, suppose this is the cell, and inside the cell you have your nucleus present. Okay, this is your nucleus, and this is your endoplasmic reticulum. Now, if any protein, let's say, is getting synthesized here, it is getting modified here, and now this protein needs to be secreted outside the cell, or it has some other role at this particular point of the cell, at some cell organelle. It needs to be transported inside the cell here, or it needs to be transported out of the cell. Always and ever, after the protein is modified and made functional into the Golgi apparatus, now this protein will exit the endoplasmic reticulum in the form of small vesicles. Okay. So now this protein will be present in a small vesicle, and this vesicle will exit from the endoplasmic reticulum. Now, once this vesicle which contains your protein, vesicle containing functional protein. Now, once it is being made exited from the endoplasmic reticulum, now always and ever this vesicle will go to where? This vesicle will go to Golgi apparatus. Okay, so now this vesicle will go and it will fuse with the Golgi apparatus and it will release its protein inside. Okay. Now see, whenever we have to post something, what do? We go to the post office, and the post office person, the postman, what does it do? It basically puts a stamp onto it. Okay. Where the parcel has to be delivered, you write the proper address of it and you put up a stamp. This is what we do. Yes, that's how the posts are being sent. You write the proper address. Yes or no? Do you write the address or no onto the envelope which you need to post? Yes. Everyone unmute yourself and say yes. I
Yes,
cannot see
ma'am.
I cannot hear my student's voice. Everyone unmute yourself. Yes.
Yes, ma'am.
Okay, Kamal and everyone. Okay. Shabranshu, yes, okay. Kevin, Samagna, Shakti, Vel, Aditi. No, Mike, ma'am. Kevin, okay. Kevin, you are always on the safer side. Okay. Now see what happens. See, what endoplasmic reticulum was doing was that endoplasmic reticulum was simply preparing the content of that parcel. Now this parcel needs to be sent outside. So what we will do, it will now go to the Golgi apparatus, and in Golgi apparatus, proper address, proper tagging, proper stamp, everything would be made onto that, so that the exact location that where this protein needs to be delivered can be understood, can be known. Okay. So now what happens, this protein will traverse through this various cisternae of the Golgi apparatus, and then it will exit from here. Okay. So now let's see this through the animation. See, these are the vesicles now containing the functional protein. So the blue colored center portion which you can see, this is the functional protein, and these are the vesicles. Okay. Now these vesicles, they have arisen from where? They have arisen from the endoplasmic reticulum. These vesicles, these small fluid round structures, these are vesicles. These vesicles have the proteins which needs to be transported at some place, either inside the cell or outside the cell. So now what will they do? They will enter into the Golgi apparatus. Now inside the Golgi apparatus, they will move from one cisternae to another cisternae like this. And while they are passing from one cisternae to another cisternae, proper tagging is taking place. Okay. So inside the Golgi apparatus, proper tagging of the proteins is taking place. And what do we mean by tagging? Tagging basically ensures the location that where this protein needs to be delivered. So proper tagging of the protein takes place for the proteins secretion. Okay. So since this protein has to be secreted or travel somewhere inside the cell, so these are being now marked. Proper address is being written on them. Okay. Now see, they are again traveling from one cisternae to another cisternae. And finally, when they move to the final cisternae, then they exit the Golgi apparatus. Okay. So now these protein final modified proteins which have the completely functional tagged protein, they will exit from the Golgi apparatus. Okay, now they are getting exited from the Golgi apparatus containing the functional protein. Now, depending on the tag, what tag the proteins were carrying, they will be transported either into the cell, inside the cell onto a particular location, or either they will go and this vesicle will fuse with the plasma membrane and the protein will be delivered outside the cell. Okay, so that's how this transport will occur. So see from here now that the Golgi apparatus, from these Golgi apparatus, these vesicles have moved out, and now either they will be transported inside the cell or secreted out of the cell. Okay, particularly the role of Golgi apparatus is mainly this, that it helps in secretions of proteins outside the cell. That is its main role. Okay, have you understood, everyone? What's the function of endoplasmic reticulum and what's the function of Golgi apparatus? Has everyone understood this? Quickly show me a thumbs up, everyone. Okay. Now, so let's see the functions. The material synthesized near the endoplasmic reticulum, they are packaged, and if they need to be dispatched at several targets inside and outside the cell, then they will pass through the Golgi apparatus. And the Golgi apparatus stores the protein, sometimes modifies the proteins and packages the proteins. Okay. So in previous slide where we proper tagging, and you can also write packaging of proteins. Proper tagging and packaging of proteins takes place for protein secretion outside the cell or inside the cell. Okay. Now, in some cases, what happens that protein modification also takes place inside the Golgi apparatus, and there particularly the simple sugars are converted into complex sugars. Okay. So along with proteins, sugar molecules are also attached inside the Golgi apparatus. And now the very, very important function of the Golgi apparatus, which had been asked so many times, is that Golgi apparatus is involved in the formation of lysosomes. Very, very important. Very extremely important. Okay. So Golgi apparatus that is involved in the formation of lysosomes. Very important. Please write down this. This is extremely important function of lysosomes. So many times a question has been asked from this. Now let's solve some questions, everyone. Everyone write your answers in the chat box. Okay, Kamalan, Aditi, Kevin, Shubhranshu, Samagna, Shaktivel. Answers, everyone. Okay. Samagna and Shubhranshu. Okay. So everyone has given the answer C. So yes, well done. The correct answer is option number C. Next. Okay, Shubhranshu, Samagna, Kamalan. Okay. Kevin. Okay. Shaktivel, Aditi. Shaktivel, you haven't given your answer yet. Okay. So now let's check the correct answer. Yes, again, very good. Option number C, Golgi apparatus. Very good. Well done, everyone. Now, let's come to lysosomes. Okay. Now, lysosomes. Lysosomes are basically membrane-bound sacs which are filled with digestive enzymes. So inside the cell, you can also see, okay? Inside the cell, you can also see several membrane-bounded vesicles which have several digestive enzymes present in it, and they will be roaming inside the cell. Okay. Inside these vesicles, you have digestive enzymes present. These structures, these membrane bound vesicles, they are called as lysosomes. Okay. Now, these enzymes which are present inside the lysosomes where they are being manufactured, so they are being manufactured at rough endoplasmic reticulum. So rough endoplasmic reticulum does what? Rough endoplasmic reticulum manufactures the enzymes. Again, enzymes are what? Enzymes are basically type of proteins only. Okay. So we already know that the rough endoplasmic reticulum is involved in the synthesis of what? In the synthesis of proteins. So those proteins, those enzymatic proteins, which will be present inside the lysosomes, they are being manufactured at the rough endoplasmic reticulum, and now those proteins, they will enter inside the Golgi apparatus, and in the Golgi apparatus they will be converted into lysosomes. Okay, so now see from here, due to various matter, now what's the need of lysosomes having these digestive enzymes? Why digestive enzymes are present inside the lysosomes? So basically, because of the various metabolic activities which are going on inside the cell, organic waste is generated. So a lot of organic waste is generated inside the cell, okay, because of all the metabolic activities which are occurring. In the cytoplasm, there would be so many reactions occurring, okay. So because of these reactions, a lot of waste will also be generated. Now, these enzymes which are present inside the lysosomes, they are capable of digesting these wastes. So lysosomes basically, they help in clearing up the wastes which have accumulated inside the cell because of the various metabolic reactions. Okay. Now, lysosomes digest the waste. Now, plant cells, they have very few lysosomes because most of the plant waste is either stored in the vacuoles or either it is secreted out in the form of some excretory substances, something oozing out from the plant surface. So, plant cells, they have very few lysosomes in comparison to animal cells. So, always remember this, that animal cells, they have more lysosomes in comparison to the plant cells. Okay, they have more lysosomes in comparison to the plant cells. Now, what happens that sometimes if a bacteria or virus, okay, so you can see from here it's a bacteria or a virus and it can harm you, okay, it is a disease causing microbe, it can harm you. Disease causing microbes, they are called as pathogens, okay, so that's one additional term for you. Pathogens are basically disease causing microbes. So if any disease causing microbe, that means any pathogen, it can be either a bacteria, it can be either a virus, or it can be either any other form as well. So now once they enter inside these cells, now what will happen? The lysosomes, they will capture this bacteria or virus, these pathogens, and it will eliminate this. So using the lysosomes digestive enzymes, these pathogens will be eliminated from the cell. Okay. Now, this lysosomes, so the first function which you have understood is that it helps in clearing of the waste. Okay. Clearing of the waste which is present inside the cell, and it also helps in clearing of the pathogens, or you can say elimination. Rather than clearing, you can use the term elimination of the pathogens from the cell. Okay. Now, apart from this, the third function of the lysosomes would be that they remove worn out cellular organelles and they remove all organic debris as well. Are everyone is writing? Everyone is taking the notes? Students, yes or no? Are you people writing? Quickly show me a thumbs up. Quickly show me a thumbs up, students. Are you people writing? Okay. Okay, everyone. Okay. Now, the third important function of lysosomes is that if any cell organelle particularly gets damaged inside the cell and whatever waste is being generated inside the cell. So you can see from here, let's say there are certain cell organelles which are now worn out. That means they are now damaged. Every cell organelle has its own lifespan inside the cell, and these cell organelles, they are being remade time to time inside the cell. So lysosomes will do what? Lysosomes, once the cell organelle completes its life cycle and completes its lifetime inside the cell and it becomes damaged and worn out, then at that time it needs to be removed out of the cell and new cell organelle will be synthesized in its place. So now you can see lysosomes, they will release the digestive enzymes. So let's say one Golgi apparatus has been damaged or one mitochondria that is also worn out. So now these lysosomes, they will secrete their digestive enzymes and they will now remove these wastes also. Okay, and this process is called as autolysis. That means they are self removing their cell organelles, and because of this property, the lysosomes, they are called as demolition squads. Now let me tell you this, exactly what do we mean by demolition. So sometime if in a city a building needs to be demolished, okay, if a building is not in a good condition, then it needs to be demolished. So similarly, when you bring JCBs or you bring a lot of labor to demolish the building, in a similar manner, lysosomes present inside the cell, they will demolish any worn out or any damaged cell organelles which are present inside the cell. And that's why they are called as demolition squads. Is it clear? Is it clear to everyone? Have you people understood? What's the function of lysosomes?
Yes, ma'am.
Okay, Shubhranshu. Okay. Now, so you can see a building is getting demolished in a similar manner. Lysosomes demolish the cell organelles. Now, another very, very important function of lysosomes. So, three functions we have already covered. Now, what happens? Sometimes the entire cell might get damaged. So, if by chance, if you get any injury, if you get any cut and bleeding starts to occur, then at that area, in that area, the cells get damaged, and those cells, okay, and even if the cells are not damaged by any injury, then each and every cell also has its limited lifespan. So that cell needs to be removed, it needs to be digested. So that's why particularly what happens, the lysosomes have the capability to even digest the whole cell. So lysosomes now, they will secrete their enzymes inside the cell, they will get burst and they will release their enzymes inside the cell, which will lead to the digestion of the entire cell. And that's why lysosomes are also called as suicidal bags of the cell. That's why they are also called as suicidal bags, because they can even digest the whole cell. Okay, is it clear? Because they can even digest the whole cell. Clear, everyone? Have you understood?
So the lysosomes will clear up the whole cell and burst.
Uh, Kamalan, repeat yourself slowly.
So when the cell is damaged fully, they will adjust by themselves, like they'll burst themselves after receiving
Yes.
all the.
Yes. So basically, there is a mechanism which is called as programmed cell death. So whenever a cell completes its lifespan, okay, then lysosomes will burst inside the cell and the entire cell will be digested. Or either in the case of sudden injury, if the cell gets damaged, then also lysosomes will burst inside the cell. Now, your question might be that, ma'am, what will lead to bursting of the lysosomes? So this process is genetically controlled. There is a whole sequence of events which take place and which eventually signals the lysosomes, okay, that now the cell is damaged and you need to burst inside the cytoplasm so that the digestive enzymes can digest the whole cell. But this entire genetic mechanism is beyond the scope of current levels discussion. So just remember that whenever a cell gets damaged or whenever a cell completes its lifespan, then in that cases, the lysosomes, they burst inside the cytoplasm and the digestive enzymes which are present inside them, they will digest and they will engulf the whole cell. Okay? Okay, Kamalan, is it clear?
Yes, ma'am.
Okay. Now, solve this question, everyone. Okay, Shubhranshu. Okay, Kamalan. Okay, Shaktivel, Aditi. Okay. Everyone else? Shubhranshu, Kamalan, Shaktivel, Aditi, Kevin, Samagna. Okay, Kevin. Okay. So now the correct answer to this would be option number B. Yes, well done, everyone. Membrane bound sacs containing digestive enzymes. Next question. Okay. Okay, Samagna, Shubhranshu, Aditi, Kamalan, okay. Shaktivel, okay. Yes, well done. The correct answer would be option number A, digestion of food particles and cellular waste. Okay. Now, next. Okay. Okay. Okay, getting everyone's answer. Shubhranshu, Kamalan, Aditi, Samagna, Shaktivel. Shaktivel, I think, is already given. Samagna? Okay. Everyone? Okay. Aditi? Aditi, what's your answer? Samagna, you meant A? You know, while I was discussing Golgi apparatus, then I had asked you to specifically note that point that lysosomes are formed where? They are formed in the Golgi apparatus. I had asked you people to highlight that point. That means you people were not paying attention. Most of you
Ma'am, I
except
did B.
Kamalan. Yes, I was just saying this that everyone else except Kamalan has answered option number A. Students, the lysosomes are formed where? In the Golgi apparatus. Okay? Please clear this point. Students, any doubts regarding this? Any doubts regarding this? Everyone, is it clear? Students, answer. Is it clear or not? Please respond. Is it clear? Have you people understood? I had asked you people to specifically note that point, okay? Now, let's conclude. So, please tell me the functions of Golgi apparatus. Everyone start writing in the chat box. Golgi apparatus, the functions of Golgi apparatus. Start writing it in the chat box. Everyone. Golgi apparatus, everyone start writing in the chat box. Okay, so they are basically what they will do, they will clear the waste out of the cell. Okay, then they will also remove dead cell organelles. They will also eliminate pathogens if they enter inside the cell, and they can also lead to digestion. They can also lead to digestion of whole cell. Okay, sorry, this is the functions of lysosomes. I'm so sorry. Lysosomes. Okay, digestion of whole cells. Now coming to the functions of Golgi apparatus. So Golgi apparatus, they will do what? They will package the proteins for their transport and secretion. And they are also involved in the formation of lysosomes. Okay? No, no, no. Aditi, it doesn't help in storage. That's just a normal function. Sometimes for meanwhile, if protein needs to be stored while it is getting packaged, the main function is that they will package the proteins for their transport and secretion out of the cell, and it is also involved in the formation of lysosomes. Okay, please correct. Yes, very good, Kamalan. They are involved in packing and packaging of proteins. Yes, very good. Okay. Now, what's next? So, in next class, we are now going to talk about mitochondria and chloroplast. Okay. So, probably I'll be taking either one of the cell organelle or both the cell organelles according to the time. So, most probably we will be discussing mitochondria. Now, see the students, the homework for today's class. So, you will go to the dashboard section. Who all are attempting test practice sessions on the platform? Tell me your proficiency. Everyone, yes. Who all are attempting tests on the platform? Raise your hands. Raise your hands, students. Who all are attempting tests on the platform? Very good, Shubhranshu. Aditi, Samagna, Kamalan.
I have problem in my dashboard. I can't log in.
Kamalan, have you contacted the customer care team and asked you to contact it?
I'll do it today, ma'am.
So you need to be active about it, na bache. Please do it on the priority. We cannot hear going on this in every class. Okay. We need to be very active on the platform. Okay. So contact customer care team. I'll also let them know today. So, but please, you yourself contact the customer care team. The number, you people already have number, contact them. Okay. Now see this, the fundamental unit of life. Okay. Now tell me your proficiency, everyone. Aditi, complete your registration and call on the number, okay, which is being given to you. Complete your registration and start practicing. So tell me your proficiency, everyone. Identify cell organelles present in a cell and plasma membrane. Plasma membrane we had covered a long time back. What's your proficiency in that? What's your proficiency, Shubhranshu? Yes, in fundamental unit of life. Understand plasma membrane in detail. And identify organelles, different organelles present in a cell. Tell me your proficiency. Students, please practice, okay? Only my this class. Yes, please, everyone, start practicing it today itself, okay? Please be a bit more efficient while working on the platform, okay? Increase your proficiency. And we are covering this topic, identify different organelles present in a cell. I want everyone's proficiency to be either 100% or at least above 95%. Okay. Please solve questions from this topic. Identify different organelles present in a cell. Okay. This is the topic which we are covering in today's class. Currently, we are going through this. So now, students, okay. So now that's all for today's class. Thank you, everyone. Bye-bye. And I'll see you next class. Till then, keep studying, keep solving questions on the platform and thoroughly and study hard. Okay. Bye-bye, everyone. Take care.